met Rasmus when I was doing my master's in biostats at Berkeley, and I audited his population genetics class, which he probably doesn't remember. So I was just kind of sitting there, and I think I was able to officially audit it. It was not illegally coming to your class, but it was a great introduction to population genetics. And then I forget how I connected with you afterwards, but I reached out when we were trying to first put together some initial inferential models for doing transmission networks. And I think I asked somebody who would be appropriate, and they mentioned you, or maybe I asked you who would be appropriate since I took your class, and you said, well, I could potentially help you with that. So I've really enjoyed working with Rasmus over the last few years. He always has really good insights, everything from basic population genetics theory to computational tricks, and the breadth of his knowledge to me is like an iceberg of which I've only seen the top bit and still been very impressed. So, and he's just such a really nice person and great to work with. So I've really enjoyed that relationship. But beyond the small work that he does with me, his focus is on statistical and population genetic analysis of genomic data in general, focusing right now, it looks like mostly on medical genetics and population genetics, including a lot of methods development, as well as general inference around key findings based on the data. And I will, without further ado, let him talk to us about what he's interested in talking about today. Thank you so much for joining us today, Rasmus.
Well, thanks so much for that introduction. That's really nice, Brian. Really appreciate it. It's been fun to work with you over the past years. That's for sure. I learned a lot. So let me see if I can share now. You should be able to see my screen now. And then I'm going to see if I can get this bar to go away. That otherwise will be in the way. Hide floating meeting controls. So I think I got that away now. So, as Brian mentioned, I work a lot in computational methods, mostly in evolutionary genomics, and I'm definitely not an epidemiologist. In fact, one of the things I'm most proud of is not trying to be an epidemiologist here doing COVID. There have been enough people like me that do mathematical modeling, but are not epidemiologists. So that I would say is the proudest moment in the career. But most of my work is really on developing methods for analyzing genomic data. And I thought if I'm going to talk to a group of epidemiologists, what might they be interested in hearing about? And I decided that maybe some of the sort of findings we are doing, some work we're doing on understanding evolution and natural selection in both pathogen and humans, and how might that fit together, this sort of relationship between selection and pathogen of humans might be something that you would be interested in and where we have some common interest. And what I'll do is sort of a little bit of a historic talk. We'll go back to sort of some of the early evidence and then talk about some recent stuff. So our focus is on these human pathogen evolutionary arms races, and there's sort of this little cartoon I show to my undergraduate students when I teach this. I talk about, it's an example of a pathogen, a virus, and how the human immune system needs to be able to recognize that virus. There's some receptors that needs to bind and so on. And then when we can recognize the virus, we can then eliminate it. But if the virus then mutates, so there's some mutations so that the immune system, can't recognize the virus, then the virus will survive. And that, of course, induces a selective pressure in pathogens, viruses, for example, to change, to avoid immune recognition. And it's then also, in turn, we think will induce pressures in the host organisms, like us humans, to try to avoid that we will also evolve over time. If there are pathogens that we don't deal with very well because of these kind of processes, then we should also evolve to address that issue. And so there's this evolutionary arms race between host and pathogens that many people have been interested in. And I think a lot of things that are going on in terms of pathogen dynamics, understanding that is really important. So what I'm talking about today is seeing this sort of the evolutionary aspects of both the host and the pathogen, and trying to see how those two relate to each other. And I'm going to talk about color samples, first of viral adaptation, first some about the role it has in the declining efficacy of influenza vaccines. So talk a bit about influenza, and then also a little bit about SARS-CoV-2. It's hard not to do that these days. And then I'm going to switch over and start talking about human adaptation. And particularly, do we see evidence of humans adapting to the pathogenic environment? It's clear that the pathogens are adapting to us, but are we also adapting to or evolutionary time to the pathogenic environment around us? And talk a little bit about the evidence we have relating to that. So the major tool that we're using here is that we're going to try to use DNA sequences or RNA sequences to look at natural selection and find the footprints of natural selection. And evolutionary biologists like to distinguish between different forms of selection, and most importantly between what we call positive selection and negative selection. So positive selection is illustrated here in the top figure. You have a population first with some individuals in the population. A new mutation then arises. If that mutation confers a fitness advantage for the individuals in the population, then with some probability, eventually all of the individuals in the population will have that mutation. So that's positive selection. Negative selection is the opposite. The new mutation is deleterious. You have lower fitness if you carry that mutation. So with high probability, eventually it'll be lost from the population. Now, if this is all that's going on, it would be quite easy to find selection. We should just find, when we see some differences between DNA sequences, that would be selection. But of course, that's not the only thing that's going on. There's also genetic drift going on in populations. So even if there's positive selection, we might actually lose the advantageous mutation. It may not go to fixation because of genetic drift. And even when there's no selection, a mutation might go to fixation in the population. So everybody carries it after a certain amount of time, just because of this process, the random process of segregation and reproduction that makes changes in frequencies of mutations through time, that eventually can drive a mutation to fixation, that is to reach a frequency of 100% in the population. So the challenge here, the computational challenge, the inferential challenge, is to figure out when are the patterns that we see, when are they a product of, for example, positive selection, and when are they a product of other processes such as genetic drift. That's sort of the main challenge, and that's has been the focus of much research in my lab on trying to develop methods that more rigorously can address these questions. And the basic two type of methods that we use to detect selection, or category, there are thousands of methods, but there are two sort of categories of methods. There are methods that use comparative data, that's from different species, or phylogenetic data. You make phylogenetic trees, and then you're trying to look at the pattern of evolution along the edges of the branches of those trees and infer if there are patterns that are compatible with certain forms of selection. The other type of data that we often use to look at or try to detect selection using is population genetic data, where you have typically, if it's humans, for example, a recombination, and you have to deal with that. There's not just one tree, there's a million different trees in the genome because of recombination. And you use a quite different set of methods to see if there are any genetic variants that you can find that might currently be under selection, for example, or have there been variants that have gone to fixation in the past. That are no longer variable in the population. And it's quite different what you can do with these type of approaches and the kind of information it gives you and which kind of selection that you can infer from these kind of approaches. I'll talk a little bit about that later on. But first talk a little bit about comparative or phylogenetic data. So the main tool that we have been using, and many other people have been using over the past several decades, is to use at non-synonymous and synonymous mutations. So remember, in protein coding regions of a genome, you can say that mutations come in two flavors: the ones that change the protein, those are the non-synonymous mutations, and then there are the synonymous mutations, the ones that don't change the protein because of the degeneracy of the genetic code. There's some mutations is from a codon that from one codon to another codon that both encode for the same amino acid. So those are the synonymous changes. We think in most organisms, the vast majority of selection is at the level of the protein, not on the synonymous changes, although there can also be some selection on synonymous changes. But because of this, we often use this ratio of the rates at which non-synonymous mutations occur and synonymous mutations occur as a proxy for selection. So without going too much detail on the statistical methods, we develop models of how mutations occur. And then we can see if the if the rate of synonymous and non-synonymous mutation is the same, or it's elevated for non-synonymous or elevated for synonymous relative to the mutational process, the background mutational process. And the interpretation of this ratio of the two, this DNDS ratio, also called omega, is that if it's less than 1, that means that non-synonymous mutations occur at a rate that is slower than you would expect if everything was neutral, if there was no natural selection. So that's negative selection. And we see that all the time. That's sort of dominating most of the organisms that we're looking at, with most selections preserving function. Negative selection is pervasive everywhere. If the NDS is equal to 1, then the rates of non-synonymous and synonymous mutation is the same when taking into account the underlying mutation rates, and there's no evidence of selection acting preferentially on non-synonymous mutation acting on at the protein level. And then what we're particularly interested in, and what is the more exciting part, is when sometimes we find that the NDS is larger than 1. That means that non-synonymous mutations occur at a rate that is faster than you would expect given the rate that you see in synonymous sites of synonymous mutations. That's the evidence of positive selection. We interpret this as selection has favored mutations that changes the protein. And so a lot of the time, that's what we're interested in. If we're interested in adaptation, if we're interested in how organisms evolve new function, that's what we're interested in, the positive selection. Okay, so detecting positive selection is not so easy because most of the time we have a lot of negative selection. If we have an important protein, there should be a lot of negative selection. So if we look at the length of the protein, we might, and look at the different amino acid residues, we might look at something like this, where most places the DNDS ratio omega is less than 1. There's negative selection acting to preserve protein function. But then there might be also some positions that have had selection for them to change, where there's been positive selection. But if we just take the average, we will, all these few positions where there might be positive selection will be swamped out by all of these other positions with negative selection. So we wouldn't see an average at the NDS ratio, a value of omega that's larger than 1. So we want methods that can address this issue. And we did that many decades ago. We did a lot of efforts on developing such methods that's been used a lot since. And the basic idea is that you use some models where you model the distribution of these NDS ratios in the domain between 0 and 1. So that's when there's negative selection or neutrality. And you then say, okay, let's assume that there's also a proportion of the mutations that are positively selected. And then you have this mixture model where you say there's some that are positive selected, some that are negative selected. And you can then test rigorously if, in fact, in your data, are there any sites that are positive selected, dealing with the multiple testing problem in sort of a principled way that gives you a lot of power still, identifying sites with these mixture models. And then when first you've done that, you can then answer questions about which sites are likely to be the ones where there's positive selection. So now you can identify individual sites under positive selection. So these type of methods have been used quite a lot over the past several decades. And if also to look at viral evolution, and some of the first good examples on viral evolution. So if we are going to ask, okay, where do we see a lot of positive selection? Some of the best examples that we have, in fact, the best examples that we have are in viruses. HIV is a very famous example. There's enormous amount of evolution within the host, as you know. HIV mutates really, really fast. There's also a lot of positive selection. Positive selection for immune evasion, and of course also drug resistance. And as you're probably familiar with, when the first HIV drugs came out, then people are very excited about them, but then they was not effective because HIV evolved various mechanisms to avoid the effect of the drug. And so people went on to make these cocktails of different drug that in the end ended up being able to control HIV much better. But that's been one of the troubles with HIV, of course, that it mutates so incredibly fast within a patient that evolution in action there makes treatment much more difficult. All right, another very famous example, of course, is influenza that we're all dealing with as we get our vaccinations or get ill every year, or maybe both. While HIV evolves at sort of within days, within a host, within weeks, the evolution in influenza is a bit slower. It's at the scale of months or years. Again, it's for immune evasion, but it's also for avoiding being a vaccine target or a target of the immune response that's being elicited by vaccines. And I'll talk a little bit more about that. And it's sort of key to understanding some of the behavior of vaccines over the past decades where there's been a reduction in vaccine efficacy. It's really to understand how this natural selection works. And we're working a bit of that. So we can use these methods. And this is an old figure. This is, back from the 90s when we were first doing that, of identifying which residues in the hemoglobin molecule of the influenza virus are under selection between different years. So maybe make big phylogenies of influenza, of the hemoglobin molecule in influenza, and then we use this method I talked about before to first of all show that there's a lot of positive selection, which is very, very strong signal of that, and also to identify what residues there are. So this is a cartoon here to the left of the hemoglobin molecule, and you have the sort of the receptor binding sites up here at the top of it, and also here you have the epitopes that interact with the host immune system. And what you see here in bubbles are residues that in this particular data set we infer to be under strong positive selection, presumably to avoid immune recognition to some degree, immune evasion. So one of the issues with influenza is, of course, that it has been that the vaccines have had a reduction in efficacy over the past years. So here's a plot from a paper I wrote together with the group of Wei Sai, one of my former postdocs, who now is a group leader at the Chinese Academy of Sciences in Beijing. And maybe first just notice the red line here. That's the vaccine efficacy of the common influenza vaccine over the past 10 years or so. And you can see there's sort of this tendency of it varies a bit, the efficacy. But there's also a tendency for it to become worse and worse, the vaccine efficacy. We want to look more at that and did various analysis to look at that. So one of the things we've been doing and that's mostly work done in a Wei Sai's group in China, is to look at this phylogenetic tree of influenza sequences from the hemoglobin molecule. So this is a tree of 32,278 sequences. And one of the things we've been doing is we've been looking at sequences that have been propagated in culture in two different ways. So there's there are multiple different ways, but there are two main ways at which you propagate viruses in the lab. One is by growing them in chicken eggs. So that's by egg passaging. And the other one is to do it in cell lines, in kidney cell lines. So there's these two different ways of doing it. An important thing to understand here is that when you make the vaccines, the strains that are being used for vaccine and the production of vaccines is being done through egg passaging. When you're going to do a large amount of vaccine production, you typically do it in eggs. And we can now then ask questions about, okay, what's the difference in the evolution of what's going on in these chicken eggs than what's going on in the rest of it that's out in circulating viruses in the human population? Are there differences in that evolutionary pattern? And what we did is we identified 12 amino acid residues that are under strong positive selection in doing egg passaging. So in the egg environment, they are on a particularly strong selection, particularly in that environment. Which then trying to do some a little bit of statistics on that, and we reduce the patterns that we see in these 12 mutations using a kind of principal component analysis. And if we plot all these strains that we have and look at which mutations are occurring in all strains, we get something like this. And we have the X strains over here, and then the clinical strains. So these are strains that have not been through X passaging. And we see that they all cluster up here. And now if we take the vaccine strains, after they've been through egg passaging, we get, these extra clusters here. There's something extra going on here. And what we can do is we can make an adaptive distance that tells us something about how far a vaccine strain has adapted in the egg from this origin of what we see in clinical strains. So we have what's called adaptive distance between what we see in other strains and then what is going on in the vaccine isolates.
Rasmus, a quick question on that.
Sure.
So looking at the upper right figure of the clinical strands, it seems like, they exhibit no variation along the first two principal components. So does that seem to you like the
this
first 2 degrees of,
This is based on just those mutations, those 12 residues that have particularly strong positive selection in the X strains.
I see.
So this in is the clinical strains, we don't see
12 to 2, not the full.
Yeah, no, it's done specifically on those ones. Yeah.
Okay, thank you.
And there are other ways this could have been done, but this sort of to extract that information that we know is, we first identify what are the amino acid residues that are really important for X-strain in X-strain evolution, and then we do the analysis just based on those to get at this. And then when we do that, what we see is the following: that at least a good amount of the variance in vaccine efficacy there's strong correlation between this adaptive distance and vaccine efficacy. So what that would suggest is that vaccine efficacy to a certain degree is predicted by how much the strain in a particular year has evolved in the eggs. And it depends on which kind of the starting strains that you have, how much they will actually adapt in the egg. Okay, how much they will go on. Some you start with certain types of strains and they will have to adapt a lot in the egg, and other ones won't adapt very much in the egg. So what's going on here with all this adaptation? Well, the H3N2 strain that we're talking about here, it originally came from birds, and there was zoonotic transfer into humans in 1968. So it originally was adapted to birds and should grow very well in eggs. And then it's been adapting, of course, to the human environment since. So that's probably one of the things that's going on here, that as the viral strain is adapting more and more to the human environment, it becomes more and more different from what it would need to grow in the egg strain. And so there's more and more adaptation going on during the egg passaging. And as there's more and more adaptation going on there, the virus that you get out after passaging, the vaccines, the finished product, is further and further away from the actual sequences you have circulating. And so there's this reduced efficacy of the vaccine due to this increased amount of adaptation during egg passaging. So that seems to be a very important feature in predicting the vaccine efficacy, that there's the amount of passaging in the eggs. So we repeated that. So these are some unpublished data here, also done in a size group that I'm collaborating with on this. And they're looking again more fine scale between 2011 and 2019. And if you take this and take more data and try to repeat this, you get this very strong, good correlation between vaccine efficacy and adaptive distance. So in the vaccine literature, most of the focus is really on the fact that you want to match the vaccine strains you start with to the viruses that you expect to be circulating next year when the vaccine comes out. And that's the sort of main focus. Can you predict what strains will be out there? And that's when you fail to predict that well, then you get reduced vaccine efficacy. So that's definitely one thing that is going on. But there seems to be a strong other effect that, in fact, years with very poor vaccine efficacy are years where you have chosen starting strains, not just one strain, it's multiple strains you start with for the production of the vaccine, that imposes more egg passage adaptation, as you grow the vaccines in egg cells. So
that adaptive distance is entirely the adaptation in the egg?
Yes, all adaptation in the egg. This adaptive distance here is all adaptation in the egg.
And to what extent are the sequences that you get after all these passagings, homogeneous, or are there many different,
they're different. They're heterogeneous. They're heterogeneous. And it's not just one sequence. It's multiple sequences. And so in different eggs, you'll get different ones that take over, right? So, but they will not all be the same.
So we're not all getting the same flu vaccine. Essentially, you and I would potentially get different sequences.
Yes, that's right. But this also tells us something that is perhaps is predictable what will happen during egg passaging. We have tons of data. We might be able to predict, in fact, the evolution that will occur during egg passaging. So what we've been trying to do work on is to categorizing the fitness landscape of the mutations. And so we can find mutations that occur together during egg passaging, more than you would expect by random, and mutations that occur less often together than what you would expect by random. So we can find positive and negative epistasis between the mutations. So what we do that is the fitness landscape. This one way of showing the fitness landscape. We basically have two main mutations that seem to drive things. So here, positive interaction is by red, and a negative interaction is by blue. There's two major mutations, and the vaccine community is very well aware of this already, that they tend to see these mutations and they affect vaccine efficacy. Mutation 194 and 186. 186 doesn't have too much of an effect in itself. Some of the other ones it's associated with do, but it's in itself doesn't have too much effect on vaccine efficacy. 194 has typically a strong reduction in the effect on the vaccine when the vaccines have that mutations in that amino acid residue. So we can think of this fitness landscape sort of like this, that there are two main peaks in the fitness landscape. And where the vaccine strains we start with end up depends on where they are on this. So if you start over here, you'll end up with 194. And if you start over here, you'll tend to end up with 186. So we can predict where you will end up. And we make machine learning methods for that. So we can, with relatively high accuracy, about 85%, predict where you will end up, which mutations you will get. And by that, also predicting vaccine efficacy, depending on the starting strain, by predicting how the evolution will occur during egg passaging. So our argument here is to people that design vaccines is that you shouldn't just concentrate on trying to predict what strains might be out there in next year. You should also choose strains that are likely to evolve during egg passaging to have mutations that are less likely to affect vaccine efficacy. And that can actually be done. We can actually predict the evolution with some accuracy. All right, so that's where we are with that right now, working on that. But I think this idea about that we have this fitness landscape, that's not just a matter of individual mutations, but how they interact, is really important for understand viral evolution in general, and can help us, because we have so much viral data, in some cases predict viral evolution. And I think this the epistatic interaction is something that has not been enough attention to, I think, so far. Okay, so there was a little bit about the viruses, about influenza. Let's go to SARS-CoV-2, which of course is on everybody's mind now. So one of the things we're interested in SARS-CoV-2, we've been working on, is trying to also do some of this stuff, look at non-synonymous and synonymous mutations. So we have a little tree here. We have the SARS-CoV-2 here, and then some of the nearest out groups. When you remove some recombination, the closest out group at the time we wrote this paper, it was not this otherwise famous RTG13 sequence, but another one, RMYNO2, that's actually closely related in most of the genome to SARS-CoV-2. We can then calculate the NDS ratios of different branches on this tree, and also within among SARS-CoV-2 sequences. And if you do that, you will find that there's been a tremendous increase in the NDS ratio in SARS-CoV-2. So if you take SARS-CoV-2 sequences and during the early part of the epidemic and compare to each other, then you get a DNDS ratio about 0.56 in average over the genome. But if you compare, say, SARS-CoV-2 to RTG13, then you would get a DNDS ratio of 0.0391. The same if you did RMYNO2, will give you about the same thing. It's about a 14-fold increase in the DNDS ratio in SARS-CoV-2 compared to what we see when we compare other related coronaviruses. All right, so why is this? There can be several different explanations. One is that you have an exponentially growing virus that can maybe be some relaxation of constraints, so that some deleterious non-synonymous mutations are being accepted because you spread so readily and rapidly that they are more likely to be accepted in this sort of early transient phase of exponential growth. That might be some of it, certainly. But another thing is almost certainly going on is that there's some positive selection, that the virus is adapting to its new host environment in various ways. And that's almost certainly going on. Now you can do the same exercises we've been doing for influenza on trying to use phylogenies to find out what are the individual sites that are under positive selection. And the one that's really been leading work on this internationally is a professor at Penn State, Sergei Kosakowski-Pond. And he's continuously updating list of what sites seems to be under positive selection. As we get more and more data, he dynamically updates list of which sites are under positive selection. And now we all think about Omicron, so I might just show one result of his. So this is, I should say, it's none of my research, it's by Sergei and his collaborators. But what they're doing is they're looking at the sites that we know are under positive or negative selection in SARS-CoV-2. And if you look at all these mutations that you have in the Omicron variant, where do they fall? And have they previously been under positive and negative selection? And this is a figure from this preprint he posted a couple of days ago. And I thought now we were talking about this stuff because I'm sitting staring at that all the time. Maybe many of you are doing similar things, wasting your time staring on, what's going on with the coronavirus. So I thought I would just talk a little bit about this because Omicron is really very, very weird. So background for the ones of you that don't know, it's comes from a lineage where the most closely related viral strains are from about February and March 2020. And it then suddenly appears out of nowhere with all these tons of mutations on it. Okay. And we look at some of the mutations. Some of them are, like, as we might expect, that they are likely adaptive mutations or ones that are relatively neutral. So if you look at, for example, up here, where the ACE2 receptor, it's mostly either positive selected or predicted neutral, that ones that are likely or possibly positive selected or neutral. But then if we look at other places in the protein, there are clusters of other mutations. So for example, here in this fusion domain here, so this is when the spike, so this is a spike protein, by the way, of course. So when the spike protein is being cleaved, so you have S1 up here and S2, there's this fusion domain down here that's important for the fusion of the virus with the host cell. And there are a bunch of mutations in here that sits in here that are otherwise under strong negative selection, very strong negative selection. So these are mutations that we, by any means, not just also these evolutionary analysis, but also when people have doing various things in cell culture and so on, are predicted to be very maladaptive. And they're all sitting together. It's not sort of something random. So you could say, oh, maybe this is just, random mutations that because they're, maybe this is something that's evolved in an immunodepressed person and it has tolerated all these deleterious mutations. But they are somewhat unlikely because they're all clustering together here. So again, it suggests maybe some epistatic interaction between these mutations, that it has jumped from one fitness peak to another fitness peak. So all these mutations that normally are deleterious individual together create some new function, a new fitness peak. So strongly suggesting that there's some completely different function of the Omicron virus, that it functionally is very, very different. It's just jumped to a completely different fitness peak, it looks like, overcome the negative fitness effect of individual mutations to find a new peak, presumably with some epistatic interactions that makes that peak adaptive. And of course, there's lots of, probably all read the news, there's lots of speculation about different symptoms and so on. We'll have to wait and see, but definitely this virus looks functionally very different from what we've seen before. All right, so that's the viruses. So next thing I wanted to do is I wanted to then talk a little bit about what we see in humans. And if we see, all this selection that we see in the viruses, the ever-changing pathogenic environment we're in, does that impose selection in humans? And in fact, if you look at the first studies where people sort of more systematically try to identify positive selection in humans, that's probably this study from Hughes in 1988. In fact, the genes that they identified there to be under positive selection is the Mhc, human Mhc genes, the HLA genes in particular, to be under having high DNDS ratios, elevated DNDS ratios. And if we do this exercise of mapping mutations on the protein, here we have an Mhc molecule, it's binding a ligand there, and we see that's all the selection in this receptor binding pocket here of the Mhc molecule. So you probably, I presume you're all familiar with the role of these Mhc genes, that they are antigen presenting genes. So they present small peptides from, well, anything they find, but also from pathogens that they present to T cells, and are really fundamental in the adaptive immune system for recognizing pathogens. And so the idea here is that as pathogen changes, you will need to have maybe different motif here. And in general, the more diversity you have here, the more different versions of this you have, the more different possible peptides you can present. And so pathogens can't avoid the immune response by not being bound to MSC molecules if you have a large enough repertoire of different MHC molecules. So it's still one of the examples we have of the strongest evidence of selection in the human genome is the MHC genes, multiple of them that show very, very strong evidence of selection. And I'll talk a little bit more about which kind of selection and so on a little bit later. So after sort of these early studies, the next thing that I would say that happened was that we got to in the early 2000s, we got genomic data. We could start comparing humans and chimpanzees. You can see this is, I dug up this old slide from like, almost 20 years ago, from when we had a different president back then that we like to make fun of. And we're all excited when we've got the first chimpanzee and human genomes, because now we could do these kind of DNDN analysis for the entire genome and figure out what are really the genes in the human genome that are under positive selection. So if we take this lineage here leading to humans from the ancestor of human chimpanzees, what are the genes that have been under positive selection during this past 5-6 million years? And when we did that almost 2 decades ago, this is a list of the genes that were enriched, the biological process categories that were enriched with genes with high DNDS ratios, that with evidence of positive selection. And right on top, we find these immunity and defense, T-cell mediated immunity genes associated with that, who are very strongly enriched with genes with high DNDS ratio, and particularly genes that are associated with the innate immune system, actually. So apobacs, for example, that play an important role in controlling RNA viruses, for example. There seems to be also enormous amount of selection here in the human host to adapt to the pathogenic environment in one way or another. And that's perhaps shouldn't be surprising if you think about what is really historically, not maybe now, but 100 years ago, or 1,000 or 10,000 years ago, what is causing people to die? What are people dying from? In an early age, so they can't reproduce and leave offspring next generation. And definitely pathogens is a very, very, very important factor there. So it should make a lot of sense that there's a lot of selection associated with how we defend ourselves against the pathogens. All right. So that's if we use comparative data. So we can give sort of an overview of what's going on, but it really is very crude in trying to pinpoint what type of selection and also selection acting on current variants, which we are particularly interested in. So has there recently been variants where selection has been acting on them? And why are we interested in that? Because these variants might be explaining differential response to pathogens today among people. Okay, so to get a question like that, of course, we need population genetic data to be able to answer those kind of questions. So the next thing that happened was, of course, we started, the community started to produce large amount of population genetic data. And then we could use various tests. And here's one of the very simple but very effective tests for trying to detect selection using a combination of comparative data, so that's between species, and also population genetic data within species. It's also based on non-synonymous and synonymous mutations. You take the nonsense and synonymous mutation within and between species, and the ratio of the two should be the same if there's no selection. It turns out that also sort of very rigorously has to be the case. And if that's not the case, it's because there's selection, and depending on the direction of deviations from equality, you can then infer whether there's positive selection or negative selection acting. And negative selection you infer is negative selection that's acting on currently segregating variants. And then you can make maps like this. We start to make big maps of selection in the human genome. Where are the genes in the human genome that are under selection? Red ones here, those are genes that are under negative selection, where there's segregating variants in the population that are currently under negative selection that we can detect. And the blue ones, they are ones under positive selection. Now, if we did a map like this today, we could, there would be many, many more genes than what we had here back in 2005. I think there was the first study that was ever done with this. It was from some early data produced by Genomics, a company that exists back then that decided to produce some population genetic data that we got our hands on. And if we ask where is the enrichment of, which processes are enriched for selection, we find defense and immunity both enriched for positive and negative selection. So again, genes associated in one way or another with defense against pathogen and immunity. Now, a very popular and common way of trying to get at identify selection is by looking at variation among multiple individuals in the world. So humans, as you might know, migrated out of Africa about 60,000 years ago. We also did earlier than that, but most people outside Africa originates from that migration and spread to the rest of the world. And we have occupied many, many different environments. And that gives us an opportunity to try to look at, okay, how is a differential selection depending on which environment you're in, including the pathogenic environment. So we and other people doing that as well have been looking at that to try to figure out, okay, various environmental factors, how important are they for selection? And the way we're doing that here is that we're trying to predict with these environmental covariants, pathogens, subsistence, that's whether you're hunter-gatherer or you're agriculture or doing fishing, whatever we're doing, and then climate variables. We're trying to predict distribution of allele frequencies with that. How much do they help with the prediction? And what we're doing here is then taking top sets of SNPs that help more and more and more with the prediction. And when we do that and find the SNPs that help the most with prediction, they're highly enriched for non-synonymous SNPs or intergenic SNPs. And that's also hold when you correct for various factors such as difference in allele frequencies and other things. What that shows us is that we're getting at something here because there's no, if it's not something functional, there's no reason for this enrichment of non-synonymous. But we also see that the enrichment for non-synonymous is much higher when we're trying to use pathogens as area and predicting distribution of allele frequency with various measures of the local pathogenic environment. The subsistence and climate, less so. And there's various ways of doing the analysis. In this particular study, no matter how we did it, we kept finding the same thing, that if we're going to look at adaptation in various ways, we find that pathogens, the pathogenic environment is what's best can predict differences. And so if you take the genes here that are selected without respect to function, ask which one is it that are best at predicting pathogen environment, we again get that enrichment and defense response to biosimilar, but immune response, and so on. They are among the top processes that are associated with this increase in prediction power of using pathogens. So that shouldn't be a surprise. It's just sort of a sanity check that what we're getting at is what we're getting at. But again, it suggests that if you're looking at the kind of how we have adapted to local environments, you might think about like whether it's cold and hot and so on. It's important, and it is. But certainly the local pathogenic environment also seems to be very, very important. Perhaps it has driven more selection than anything else. Then we can ask which kind of pathogens. And when we did this back in 2011, what we found was that, in fact, what is most important for prediction power are helminths, so worms. It could be because we're not so good at describing the historical local pathogenic environment, for example, for viruses. But we find helminths have had a particularly strong effect on the prediction power, so seem to be particularly important in terms of the pathogenic environment that we respond to evolutionarily.
Rasmus, could it also be, I mean, maybe this is inherent in what you said, but that helminths are more geographically stable in terms of the,
exactly.
Okay.
So that's possible why that if we could, if virus is not so geographically stable, although they may have been more so, you know, when there was less mobility of humans as well. But so there is that caveat to it, that this is only as good as your ability to actually describe the historical environments. And the pathogenic environments, of course, historical environments are described with great uncertainty. So there is that caveat. I completely agree. So that's one way to look at it. So another way to look at it is just to ask, okay, what genes do we see that there's a lot of selection that has differentiated humans? Where are the genetic variants in humans between different groups of humans where there's been a lot of selection acting on differentiating humans? And we developed recently lots of different methods for that. There are other methods out there, but in very brief, what we do is we use statistical model of the genetic variation among different groups of humans, where we can model them statistically using, if you're familiar with structured mixture models similar to that. Based on that, we can make trees that are sort of a statistical model of the covariance among different groups, and also describe the allele frequencies in the groups. And then we can ask, are there particular genes where this model fits particularly poorly because certain groups are just more differentiated in SNPs and genetic variants in those genes than our genomic background model would suggest. So it's a way of dealing with all kind of confounding factors. So we built a model of the genomic background of the relationship among individuals, different places in the world. And then we can ask specific questions based on the tree about different ancestral population groups, if there's been selection in those groups. And the way we do that is, maybe I don't really have time to go through that in too much detail, but you can basically, you ask if there's an elongation of a branch in a associated with a particular SNP. So a SNP has changed allele frequency more. So to describe the pattern in that SNP, you need a longer branch for a particular population or sets of populations. And we can then do likely ratio tests and other things to then test whether a model with this increased change in allele frequency change in one population fits the data better. So we're basically looking for individual genetic variants that change specifically in one population a lot in its allele frequency, much more than what you predict given what you see in the rest of the genome. And this is a list, for example, we come up then with a list of genes for this. This is a list of genes if you do this for Europeans and compare to other groups. And what are these top genes then? If you look at the top genes, there's a ranked list. These are the top genes here. Okay, so the genes that we find here are all of these genes are in fact associated with hair color, skin pigmentation, or eye color. And it's something that has been known for a long time, that they really, really stand out. So if you look at, we can make a plot like this, where on the x-axis, you have the genome-wide FST. That's a measure of how genetically differentiated two populations are. So each dot here is a pair of groups of individuals, and you can call them populations if you want, different places in the world, and how genetically differentiated they are. And then on the y-axis, it's the maximum frequency difference between any gene in these, or any SNPs in these populations. And what we find here is that what this shows us is that sort of a pattern. Of course, the maximal difference correlates with the average difference, but then there's some outliers here. And those are all these skin pigmentation, hair pigmentation genes, eye pigmentation genes. And what this tells us is that those genes are the ones where we are the most differentiated from each other, by far, by anything else. And we think we understand why. It's mostly has to do with UV radiation. If you're in an environment with a lot of UV exposure, you want to have a lot of pigmentation to protect yourself against the cell damage from UV exposure. If you are in an environment where there's not very much UV exposure, you could suffer from vitamin D deficiency. And so you want to have less skin pigmentation. So most of the genes that show really strong evidence of differentiation between populations are associated with this. And it's a little bit interesting because it also tells us that if we use skin pigmentation as a way to look at the world, we're greatly overestimating how different we are from each other. They're very, very unusual. We are genetically much more similar to each other than you might expect, given what you see regarding hair pigmentation and skin pigmentation. This gene here that's also here, by the way, it's not a gene we worked on, which has to do with how Tibetans have adapted to high altitude. In any case, these are the top genes. We kind of know the story. There's another here, in one gene we know well here. This has to do with lactase, so avoidance of lactose intolerance, adaptation to being able to drink milk as adults, in another famous case. But then we start to get immune-related genes. For example, tolerite-like receptor 1 comes in here. That is a gene that is very, very important in the innate immune system. And we still don't quite understand why has there been so much selection for new variant in Europeans in this particular gene. It's sort of unknown. What is that associated with that variant? All right. So right now I talked about mostly, and I know I probably need to finish up soon. So I talked about mostly about positive selection and negative selection, and that's sort of one way to categorize selection. But we also often talk about balanced selection. Balancing selection is selection not like positive negative selection, but selection that maintains genetic variability of the population. So instead of one mutation going to fixation or loss, the selection acts to maintain variability. So you keep having genetic variability in the population. Okay, so we worked a lot on this type of selection as well, trying to find that. And what we're doing is we're modeling basically the genetic pattern you would expect to see under such selection. We have models of how natural selection should act in these cases. And then we can look at the genome and say, okay, if the balancing selection affecting a particular SNPs, how should the whole pattern of genetic variability look in the genome around that SNP? And then we can query the entire genome and try to figure out where's the evidence of balancing selection. Now, it turns out in humans, there's not that much evidence for a lot of balancing selection in many places, but where we do see it, that tends to be also immune and defense-related genes. And particularly, and I'm coming back to that now, the Mhc genes. Okay, so that's by far where we find the most balancing selection. So this is the region where you have all of the class 1 Mhc genes. So you have several different clusters here where there's extremely strong evidence of balancing selection, much more than what you see anywhere else in the genome. And so it's a very old discussion now in why is there so much? All this balancing selection has been known for decades that it's there, but it really, really still stands out. So why is all that? Why is it balancing selection? We saw in some other cases, we saw that we have these mutations that go to fixation all the time. In the MHC, that doesn't seem to happen. Instead, we have all this variability maintained. And one possible explanation, classical explanation for it, is that that it's over dominance, that it's better to be heterozygous. If you're heterozygous, you can bind more different possible pathogens or peptides from pathogens. You can have a more wider immune response that can target many more different pathogens. So there's selection for increased heterozygosity, and that's what causes balancing selection. Certainly, there are other factors affecting it well, but that might be part of the story. We still think that about the Mhc genes and why they are so incredibly strong balancing selection in humans. Okay, so just to sum up a little bit, some of the sort of different ideas. We have selection at different timescales that might explain some of the different dynamics we see. We have viruses, ultra fast evolving, HIV really, really fast, faster than influenza. That's about 3 to 4 times faster than SARS-CoV-2. Bacteria, they also evolve fast, but much slower than the viruses. Then we have eukaryotic pathogens like helminths and plasmodium. They are slow compared to these viruses, and bacteria are slow evolving. And then ourselves, we are also slow compared to the rest. So the timescales and this interaction between the pathogens and the host, us being the host, I have to consider what's going on at different timescales. And I think that can explain some of the different patterns that we see. We see these genes in the innate immune system, but in particular are the ones that are under the strongest positive selection in terms of high DNDS comparing to the chimpanzee, for example. So there's where you have new mutations that go to fixation again and again. And maybe one way to think about it is that the innate immune system has no chance of really co-evolving with a virus because the virus evolves so much faster. There's no real co-evolution. But at some point when the pathogenic environment, the factors in the innate immune system are completely irrelevant, and then there's a mutation that then suddenly is relevant, then that mutation will go to fixation. So there's not sort of a dynamic segregating mutations that are interaction between the pathogens and the host. They are so likely. It's more like you have a complete change of the repertoire. Sorry?
Sorry.
Okay, no problem. Then if you look at ongoing selection in humans that's causing population differentiation, it seems mostly be related to the eukaryotic pathogens, helminths. We also have many cases with Plasmodium, Duffy, beta-globin, and so on, where we see that. So they evolve so much slower that now you can have this interaction where the pathogen is evolving, the human is evolving, and we get segregating human variation that's sort of co-evolving together with the pathogens, because now we have timescales that are more comparable. And then we have some other genes like the Mhc genes that are then end up being under overdominance. Again, the pathogenic environment changes very, very fast, and you just need to keep a massive repertoire all of the time. So the result of that is a form of balancing selection. So I think these different timescales can explain a little bit of at which different organisms evolve, why we see slightly different patterns in different parts of our immune and defense system. All right, so I will end there. I think I'm a little over time here. Acknowledge some of the collaborators, particularly the Wei Tsai and his group in the Chinese Academy of Sciences, Pipes Hong Rang, who work with me on some of the SARS-CoV-2 evolution stuff. They're postdocs in my group right now. Matteo Fumagalli, who former postdoc who did the selection using environmental covariates, Mike DiGiorgio, who worked on balancing selection, a former postdoc, also Jay Ching, another former postdoc who worked on these methods on selection based on population differentiation, and all other collaborators that I haven't mentioned. And you, of course, thank you for listening to me today.
Great, thank you, Rasmus. That was, you covered quite a lot of material.
I know. I thought it was more fun with like a big overview than too many nerdy little specific details. So I gave you that. Hopefully you find it entertaining.
Yeah, no, it was really interesting. There was a question in the chat from Josh back to the virus work where he asked, is Josh still on the call? He may have had to drop off, but it's an interesting question. Just to what extent jumps seen in Omicron relative to wild type also appear in evolution of other viruses?
I mean, we see that all the time, of course, zoonosis all the time. I mean, in influenza, in coronaviruses and so on. So I think that's what we expect to see, and that's going to continue to happen, right? So that is not unexpected. I think with Omicron, it's just just a little bit of speculation with that. There's two hypotheses, right, where that comes from. One is that it was, as you know, they have a very high proportion of HIV infected in people in South Africa, also people with out treatment, and so it's an immune suppressed person where it's sort of been there latent for a year and a half, almost 2 years, and have been allowed to evolve all these changes. Another one is that there was a reverse zoonosis and then a zoonosis, so it's been an hippopotamus or something like that there. And I don't think we know which of these two hypotheses is true, but I think that might be might have been the second zoonosis from SARS-CoV-2 that got us here potentially. Anyway,
Jimmy, you have your hand up.
Oh, hey, Rasmus, that was a phenomenal talk. I just have two quick questions. One is, we've been studying ERAP2. I would love to hear your thoughts on sort of if you've analyzed that locus a little bit deeper.
Yeah, I haven't worked on ERAP2 for a long time, so I actually have nothing new to contribute on that. But I'd love to hear more about your recent work on that.
Yeah, it might be a longer discussion than this call, but I'm happy to follow up on that. Then a second question is I was really sort of excited about your sort of comment about epistasis. Given some of the newer work using deep learning models to functionalize individual protein coding variants, do you think that there's an opportunity to go beyond sort of like DN and DS ratios to just, in basic model for detecting positive selection or native selection?
Yeah, I mean, I think this issue about epistasis, as I said, I think is important. And so the question is how, have models for detecting that. And I should say, I didn't really go through how we're actually doing this on when we do the epistasis on the tree. The way we do that is we have these methods for stochastic mapping of mutations. So we map where the mutations are, but if you just do one mapping with parsimony or something like that, we'll get huge biases. So we do it stochastically with models of molecular evolution, and then stochastically we sample ways that mutations could have occurred on the tree to create what we see today. And based on that, we then can make these inferences about co-occurrence and so on. And I could imagine that something that would be better to work on, if let's say you want to use machine learning methods to try to infer some of these things, is actually these inferred patterns of mutations rather than raw sequences. That sort of you made it a little bit easier to go that way. But I could imagine taking sort of a convolutional neural, make back of all the mutation, all the different sites that occurred, and then throw a convolutional neural network on it. And I think there's enough data for the influenza virus that could give something interesting. The only thing I would say about that is that mechanistic interpretation of it becomes harder, right? I mean, one advantage of these kind of models I'm talking about is that there's a specific interpretation in terms of the evolution, that when first you throw it in the deep learning black box, interpretability is suffering afterwards. But for prediction, for example, for this thing we're doing with prediction on how it will evolve, I certainly think that that's probably the way to go. Yeah.
Great. Any other questions particularly from folks outside the epicenter before we have a smaller conversation? All right. Well, if that's the case, thanks everybody for joining. And Rasmus, do you have the link for the second?
I think maybe it was in an email to me, right?
You know what?
Yeah, I have that. I have the link. Yeah.
Perfect.
Stay here because everybody's still here, right?
I think so. Yeah,
I think we can stay here. There's.
Yeah, I think everyone else has dropped off.
Yeah.
Okay.
Perfect.
I actually wondered if you would follow up. Do you see in the chat the first question from Josh Batson?
Okay, maybe
that's the one I asked.
Yeah, maybe I didn't answer it properly. Okay, let me read it. If zoonosis was in 1968 and adaptation to human hosts occurs since then, why would we be seeing this decline ever due to X passage only recently? I mean, that's a good question. Most of the adaptations should have been occurring early, right? So you would expect that to occur. So I think that's an open question why we see it so much. I mean, it might take a long, the virus has perhaps been adapting in many different ways to, the human system, and there's been all type of adaptation. And now it's really the adaptation that's been imposed maybe by the vaccines. So another element of this is that the virus, if the virus can evolve to be maximally different from the type of mutations that occur during X passaging, then it can avoid much of the effect of the vaccine. And so that might sort of be the tail of its, now it's selection that's really imposed by the vaccinations that we're seeing now, not just the adaptation to the human environment become more different but it's specifically selection imposed by the vaccinations. And how can you evolve to become generally, unlikely to be targeted by vaccinations? Well, you do that by being maximally different from the mutations that unintentionally always are in the vaccines, the ones imposed by egg passaging. So I think that makes a lot of sense, that perhaps that's what's going on. It's sort of a secondary effect we see now that we're imposing ourselves with the vaccinations.
Great. Maybe we could just do quick introductions. I think other than Max and Teresa, I'm not sure to what extent you've met other people. Isabel, do you want to just quickly introduce yourself, and then we can?
Yeah, I think we met briefly a long time ago in one call with Ina. But yeah, I'm Isabel Rodriguez. I'm an assistant professor and co-lead the epicenter with Brian, and I do research mostly in modeling and epidemiology of infectious diseases with a heavy focus on arboviruses and vector-borne diseases, including malaria and arboviruses.
It's medium.
Yeah. Andres, maybe?
Well, I'm Andres and I'm a postdoc, and I'm going to talk one-on-one with you in an hour, right?
Yes, that's right.
Great. Marco?
Hey, I'm Marco. I'm also a postdoc. I in a previous life, I guess about a year ago, I was working on phylogenetic methods and trying to quantify basically fitness effects of continuous traits of interest and their evolution at multiple scales. So there's a lot of maybe some sort of intersection in our worlds. And yeah, so thank you so much for the talk and for being here.
Good to meet you. Yeah.
Joe?
Hi, I'm a PhD student in Brian's lab, and I look at serological markers of exposure to malaria. Yeah.
Good to meet you.
Great. Cece?
Hi, I'm Cece. I'm a SRA. I'm a research tech for Brian's lab. I'm just working assisting other lab members. Yeah.
Great. Sophie?
Hi, I'm Sophie, and I'm the other SRA.
Hi.
Saki?
Hi, I'm a postdoc. I'm an infectious disease epidemiologist and modeler working closely with Isabel and Brian. And I think a lot about seroepi and kind of like how different multiplex technologies could tell us about population epidemiology.
Cool. Hi. Good to meet you.
Sophia, and